this is two strategies that we're pursuing to do project-based learning. One of those is a CRO. We call that a Novo Bio. That's the name of our contract research organization. And that's an example of a centralized type of project where projects come into a central resource lab where there's people and equipment, and we do work with people. The other part that we're pursuing with support from the National Science Foundation is a decentralized project. And I'll try to talk about that just for a second at the very end. In our institution, Caldwell Community College, we have a Novo Bio operates sort of as a separate program within our biotech program. And it has 3 people that have been hired specifically to work in this program. We have a director, an assistant director, and a research technician. And these guys do not teach. They have been hired specifically as a professional staff, and they mentor students on research projects. They go out, they recruit projects in from companies, and we bring those projects to our lab, and the students do work on them. We do have a full contract mechanism in place. We've developed paperwork for that provides assurance to the companies in terms of intellectual property, as well as addresses the issue of non-disclosure, which is quite a sticky issue because we have high school students that work in our facility, and that makes it an interesting mix of projects for companies. Active projects, and I am going to go through this really quickly because I think you get the idea there that we are bringing, there's projects everywhere, and companies are one source of those. As you heard from other people, there's projects elsewhere as well. But our students are active in doing protein expression, purification, cell culture, cloning, various kinds of species identification methods for plants and funguses, and all kinds of real-time PCR. And we have, at this point, about 30 students that range from high school students to our college students to, Tammy mentioned earlier, our university partner. And they send their students to us as well because they don't have a research function at that university, teaching university. So their students will come to us. They've now started at a Novo Bio as well at their institution modeled on ours, so they do have that now. And a problem that emerged when we were sort of setting this thing up was the fact that we had diverse student groups that were trying to work together in teams. And that sounds like something everybody is probably interested in. They want to see teamwork. And it was difficult because students would come in, they would have laboratory notebooks, of course, where they're keeping their records. And the idea was they would open a notebook and try to digest what had happened before and pick up at the end. If any of you have ever worked in a research lab and you're looking at somebody else's notebook, you might understand that that's a real challenge. So one of the things that we implemented to sort of facilitate student communication and teamwork was a really simple thing. We use Google Groups, where students now post notes on an ongoing basis throughout the day when they do work in the lab, and then everybody gets updated with that. So I'm going to skip past this. So what I thought I would do is just, I get these in my email on a running basis during the day. And since, I was trying to rework this thing a little bit, I thought I would just show you a few of these to illustrate the kind of things that come out of project-based learning, particularly in the CRO model as we're practicing it, and the fact that it certainly does lead to some interesting things. What we weren't able to do originally was catalog this. We weren't able to capture this aspect of the project-based learning. We would have project reports at the end of the day. We would take those to the companies and we could say, oh, look, students are working on projects. Isn't that great? But we didn't have a way to show the student-to-student interaction. And we heard this morning about how these sort of projects are great places to assess these soft skills that companies say they want. But I think this is a really interesting way because we're capturing once a transcript of what happens as students communicate with each other over the course of a project. So for example, I like this one. This is a problem-solving one. This is for a project from a company called Mudra, which is one of these small startups, as we've heard, are excellent sources for projects. I like this because he starts off and he's talking to the members of his team. This doesn't seem right at all. I'll have to check it out. I'm thinking either in this read the latter or the new DNA is different. Today, Crystal purified this construct and ran this on gel. So already you see right there these elements that they're talking to each other about. I'm not sure what's going on here when you think about this and compared to what our assumptions were about the work. And then he goes on, of course, to just describe what else is going on with the project. And this helps the next person come in and be ready to start the work. Teamwork. Let's see. I like this piece down below here is what I really like, this little bit right at the bottom. So I'm just going to read that little bit. So there's this long description here of various colonies and genes that were analyzed with PCR. And he ends by saying, we got great bands for all 4 of the genes and set to use using a plug and smash method on Monday. Great work, guys. And I like that because this guy, this is Eric, he was in my 1010 class, my entry-level class last spring. And he's already moved up through our Nova Bio program into leading a team of students. So he's got about 4 students working for him. And you see he's developing this characteristic of praising people for good work. And you see that emerge from this conversation. I never would have been able to capture that just by looking at a laboratory notebook. So I'm really proud of that aspect. Community. This one's just funny because Darden, who started with us as a high school student, is also now in a position of leadership and is leading a group of students on a baculovirus project. I like that he was sick for a week, and he posts, Chelsea and Miguel have won a free coupon for 1 meal from Subway, courtesy of me. I owe you guys for getting things done while I was sick. And so you see, there's a community here where they're communicating as though they would communicate with friends, and that's something we haven't set out to create. We haven't set out to create cohorts. But you see that happening naturally, and this sort of tool captures the fact that they're comfortable with each other, they're talking to each other about data and scientific communication, but also communicating as colleagues would communicate. And I really like that about this mechanism. And finally, I just put this one up. I think it may be apparent, but today I ran the gels from the PCR Ellen ran using the first set of primers. The NTR products did not show the GAPDH at a very distinct band at the right size in 3 of the 5 lanes. I extracted the 3 lanes, and so on. And this is being posted by Bree. And I just want to say that I wasn't able to communicate with people like this when I started graduate school. Okay, this took time for me to develop in graduate school. Brianna is a second-year community college student who's now moving into the 4-year program. And so you see the level of communication that she's able to use in communicating with her team. And I don't think they appreciate the fact that they're communicating at a level that's really approaching graduate level types of communication. And this is cool because I can now take this as a transcript and we can present that to somebody that wants to hire them, or this can go into an application in graduate school or other things, if appropriate. But it certainly shows something about their capacity to work in teams and to communicate with each other. And this is all facilitated, of course, by the fact that we have these companies providing projects that the students then collaborate to work on together. And you'll notice, nowhere in here did you see Adam or Mary or Alejandro mentioned, who are the 3 major staff members that are sort of in the background. And that's something we really like. They're in the background as a resource when necessary. So I did want to mention very briefly that, since my time is almost up and the day is at the end, I do want to mention that the Innova Bio project churned in my brain for a long time. The fact that we were able to go out and get projects and bring them into our lab in this central resource model. We have a lot of high schools that we work with. High school programs are really important for us. And one of the things that bothered me was that it was never a way to really take the projects out to them. We really struggled to think of ways to do that. So, like many people, my thoughts turned to genomics. And we have now created, with a STEP grant, support from the National Science Foundation, we are now serving as the company, so to speak, that needs to contract out projects. So either Solid Community College or our partner, Utah Valley University, now subcontracts to various high schools parts of a genomic research project that we have undertaken with a collaborator from Westminster College. And the project is basically Dr. Bonnie Baxter. She's provided for us an isolate from the Great Salt Lake. So we have a halophile called Halorubrum salsolis. It was named by this young lady here. She's 9 years old. And it's a new species. It's never been examined before. She's interested in it because it has some interesting photobiology properties. And we went to her and said, how would you like to have the genome done? Because we have a group, or at least some gene discovery done. Because we didn't intend for it to be a genome project at the time. And she said, that's great. And so where we are now is we've gone through 1 year of our first year of our STEP grant. And in doing that, we've worked with all of our high school teachers. These are all of our high school teachers we brought in during the summer. We call it our Summer Science Institute. And they are working through how to convert technical cloning manual, basically, into a high school teaching document. So they spent the whole summer, or spent a week of the summer with this. That's what they're doing. They're basically taking the documents that we developed through our CRO, how to make libraries and so on, and they're now cutting it to pieces and making it fit into their high school day. So that's what they're working on right here. Some of them are working harder than others. Steve over here is always a good one. So, in fact, I've got another picture of him asleep with that thing on his chest right there. So, in any case, this went on for a week. And then we took them, we talked to them about the theory behind sequencing and how it actually happens. We have them in the lab actually going through the protocol that they've helped create And now the job for them is to go out into their classrooms during the course of the year, which is right now, and do this work with their students. So we've subcontracted little pieces of this genomic project out, and we now have over 350,000 bases, unexpectedly, of genomic sequence for this organism that's never been looked at before. And I like to tell all my students that, next time you go to a party, you be sure to tell them that, when they're complaining about how boring school was, that you got up today and discovered that something nobody had ever seen in the history of the universe, and see what they say. And usually that gets some eyebrows raised. And that's pretty impressive for a 40-year-old guy to go talk to a bunch of teenagers and get them to be interested in a science project. So we do have support for them, so we've created also a website where basically we recognize these are high school teachers, and for all the reasons we heard about this morning with No Child Left Behind, the mandates, various things, we knew that this was not something we just turn them loose and say go do it. So we created centralized curriculum documents, which are here. They can also go on, and there's recipes, and we've also built into our grant support for them to request the reagents, because there's no way with their budgets they're going to be able to build that within their high school. So they can request reagents through a drop-down checklist, and we'll bring them a package, and that also helps to keep them moving along the way on the project. This is in line with the sequence that we've gotten against another related species. So the contract research organization for us has served the purpose of providing project-based learning. It's facilitating very interesting development of soft skills, we think. We're beginning to see that emerge in our Google Groups discussion tools. And it also served as an inspiration for how we might serve as an outsourcer of projects to our high school programs.